introduce him. He is the Boya Distinguished Professor and Chair of Chemical Biology at Peking University. Dr. Chen obtained his PhD in chemistry at the University of Chicago in 2007 and finished postdoctoral training with Professor Peter Schultz at the Scripps Research Institute in 2009. He joined the Department of Chemical Biology at Peking University in 2009 as an investigator, working his way up to professor in 2014 and then chairman of the department in 2015, and is now director of the Synthetic and Functional Biomolecule Center at Peking University. In 2020, he became director of the Division of Chemical Biology of the Chinese Chemical Society, and in 2022, assumed the role of executive editor of ACS Chemical Biology. His lab is a leading group in developing bioorthogonal cleavage reactions for chemical-controlled protein activation and proteomic profiling in living systems, leading to the creation of a new direction in bioorthogonal chemistry for gain-of-function study of proteins in their living systems. We are most grateful to have you here today, Dr. Chen. Please take it away.
Thank you. Thank you, Rick, for the nice introduction, and thank you, Ryan, for the invitation. Now let me share my screen. Can you see my screen now?
Yes, we can see it.
Okay, all right. Yeah, this is really an exciting opportunity to share the most recent progress in my lab. And I really appreciate the opportunity, and good morning and good afternoon. And this is now at 10 p.m. In China, but this is really a fun meeting. Today, I'd like to talk about how we leverage the genetic code expansion technique to do chemical reaction, particularly bioorthogonal reactions in living systems with high spatial, temporal, or spatial and temporal resolution. So let me start with a key question we are interested in. How are the proteome organized inside a cell, spatial and temporally, which is essential for life? We all know that the central dogma is dictating theory for the life processes. But after the DNA got encoded as a protein, there's actually a lot of post-translational events occurs on protein inside a cell. So after the Human Genome Project, we are surprised to know that there's only 20,000 genes, which is for human, which is about the same number as a mouse. So the complexity of the human is really actually coming from the complex proteoform inside of cell. And this is generated with, as I mentioned, lots of post-translational events and the spatial subcellular activations of the proteome. This is actually one of the 125 key scientific questions listed by Science magazine, as how are biomolecules organized in cells to function orderly and effectively. To study this question, we have to develop tools inside a cell so we can understand the role of many proteins in situ. To this end, we need genetically encoded tools for protein study. Currently, we are pretty, we are having many tools to study proteins in vitro, including biochemistry strategies as well as structural biology studies. However, in order to study a protein inside a cell with high spatial and temporal specificity, we are lacking tools. Small molecules, many of them actually lack specificity, whereas the mutation or overexpression, this kind of traditional molecular biology tools lack the desired spatial temporal specificity. As a chemist, we know that to conduct chemical reactions inside a cell is a long pursued goal, as evidenced by the most recent Nobel Prize in Chemistry that awarded to these three laureates in recognition of their contribution to the click chemistry as well as the bioorthogonal reactions. However, we know that all these bioorthogonal reaction handles, for example, alkyne, azide, as well as all the functional groups I shown here, are not encoded as side chain proteins. So in order to perform a specific reaction on a protein of interest, we need a genetic code expansion technique. I'm sure the audience here are familiar with the genetic code expansion technique. So I will skip the details. If you want to see the details of protocol, you can see many review or protocol papers, particularly from the pioneering group of Professor Peter Schultz's group at the Scripps Research Institute. But to make a long story short, this technique can allow you to incorporate an unnatural amino acid. The side chain will be different from the canonical 20 that with the orthogonally engineered amino acid synthetase and tRNA, you can incorporate this amino acid into the protein. And so the protein of interest can bear a site-specific incorporated unnatural amino acid. This can occur inside of prokaryotic as well as the eukaryotic cells. For example, with this technique, you can incorporate alkyne into a protein. And then if you have azide containing amino fluorophore, you can do specific protein labeling on the cell surface as well as inside a cell. I call this genetically encoded click reaction. And this will allow you to do the protein labeling. Right now, there's a growing list of unnatural amino acids that can be incorporated in this through this method. And the living species that can accommodate this kind of technique are also increasing from the bacterial mammalian cells to multicellular organisms and even the mouse. So let me give you two examples that we are currently conducting in our lab through this kind of click reaction or more generally bond formation reaction. The first example is we site-specifically introduced amino acid into the protein for to generate a fluorescent sensor for the voltage. Currently, they're either the fluorescent protein-based or the small molecule-based method that either are have a low brightness or have a high non-specificity. Alternatively, we use a genetically encoded method where this TCO, transcyclooctane, can be site-specifically introduced into a rhodopsin. This is a GPCR that has a voltage-sensitive protonation state with a slight color change. And then with this insertion of the TCO, we can introduce a tyrosine containing fluorophore. Through this inverse electron Diryzado reaction, this fluorophore can be site-specifically introduced on a specific on an extracellular loop, so that this fluorophore can form a Fred pair with this internal fluorophore of rhodopsin. And then this will the fluorescent change will reflect the voltage change of membrane protein. Therefore, it can record the voltage change as a sensor. We call it hybrid voltage indicator. And this have been realized by site-specific and genetically encoded bioorthogonal chemistry. And then we can visualize neural cells activity that have been traditionally using this patch clamp strategy. And now with this fluorophore, you can visualize with multiple channels, you can monitor multiple behaviors of a neural cell in situ. And we are currently moving this towards more live cell or live animal settings. Another example, as I mentioned, the bond formation is the cross-linking, where this diazorin containing photocross linkers can be site-specific introduced into protein, and then you can covalently capture its partner protein for mass spec study. We have been developing this for almost a decade, and the most recent tool is actually we generate the diazorin on the backbone rather than the side chain of a lysine. Therefore, we can leave the lysine this space for further PTM containing unnatural amino acid. So we call this a bifunctional unnatural amino acid, where it containing a PTM on the end of the side chain, whereas on the backbone it has a diazorin. So that this will once site-specifically incorporated into a protein, for example, histone on the chromatin, it can mimic a site-specific histone modification. For example, histone lysine acetylation or acylation. And meanwhile, this diazorin group can form the covalent bond to capture the proteins that can recognizing this specific PTM. we show that this strategy works nicely inside a cell, and in particular, once incorporated, it will actually assemble into the intact chromatin as a histone protein. Therefore, it can study the real inside a cell as well as a chromatin context-based protein interactions. And with this method, we can actually understand how this chromatin mark, for example, lysine isolation, define the protein interactions, as well as this mark will define the genomic location of these modifications. So basically, this strategy can help us to link these modifications to genome distribution, as well as the spatially organized proteome intactome. So we call this strategy cytomics, single site resolved multiple omics. And this is one of the example where this 3D, we call this chromatinized 3D proteome, because this PTM organized in 3 dimensions by the chromatin, and on the different chromatin, it has a different interaction proteome, and this method will capture this in situ. So that's we are currently expanding this tool to other PTMs that occurs on histone proteins. Okay, those are all the bond formation reactions. As Rick mentioned, that we're also leading the development of bioorthogonal cleavage or bond cleavage reactions for applications for including protein activations. So we hope that to develop a tool that can study the protein activity in situ in living cell. This is a challenge because there are so many proteins. For example, signal transduction enzymes inside a cell, just like a traffic in the whole city. If you want to study each intersection's traffic light, we will turn it off. We hope to turn it off and later turn it on. So basically, we want to add a switch onto each of the protein. And then when we develop a cleavable reaction, we will turn on the protein activity one at a time. So this is a gain-of-function study of individual proteins. But the challenge is to study that inside of cell, just like you lock a specific protein and later on you activate the protein and then follow its activity. This has been realized by one hand by the bioorthogonal, we call cleavage reactions, where the presence of the catalyst or the small molecule or the light can trigger the cleavage or deprotection of a reaction on a amino acid side chain. And once we incorporate the amino acid into the protein, we can log the protein activity. For example, we use the CAGE, the lysine, to block the enzyme activity of kinase. Once we activate it, it will rescue the ATP binding and the phosphorylation, and then we can study the phosphorylation events of a specific kinase. Because this lysine is highly conserved in kinases, we can study virtually any kinase of interest inside a cell. We show this is indeed the case, and our strategy works on both the serine threonine kinase, the MAC1 FAK, as well as the tyrosine kinase SARC. And so one particular case is that we hope to study this SARC activation, tyrosine kinase activation, because this is a much lower abundant kinase substrates, and also as the tyrosine phosphorylation is highly dynamic. So we build a pipeline that we can in situ activate the kinase inside a cell, and then we follow up a systematic phosphoproteomics to capture the phosphorylation events activated by this specific kinase. And the question we hope to address is what are the native substrates of a given kinase, and in particular, how are the signaling rewired upon oncogenic mutation occurs on these kinases. That we know that a lot of oncogenic mutation will cause lung cancer as well as other cancers. The problem to address these questions is that these occurred very fast inside a cell within second or minute time scale. That's rendering the current traditional methods like overexpression too slow to study this kind of highly dynamic questions. So to address this, we use photo decaging. The reason that this is really faster than chemical decaging. And we show that upon the activation of a given kinase, we indeed can differentiate the direct versus indirect substrates of the phosphorylation events, but this is within a half-minute temporal scale. If without this high temporal resolution, for example, if you do the phosphoproteomic study after 1 minute or longer, you're now able to differentiate direct versus indirect substrates, which indeed showing that this is a very rapid event. So we activate a specific oncogenic Src kinase. This kinase oncogenic mutation will cause overwhelming phosphorylation inside a cell. But when we did this temporally chase that we call the pulse chasing kinase activation strategy, we can capture direct substrates of these kinases. And we found one specific phosphorylation on a ubiquitin enzyme, UBA1. This is a U1 ligase. A specific phosphorylation of this UBA1 causes a significant suppression of UBA1's activity. And further mechanistic study showing that Src activation will phosphorylate UBA1, which will cause the depression of its ubiquitination activity, and thus accumulation of lots of kinases that's supposed to be ubiquitinated and degraded. This aberrant accumulation will further feed forward the whole phosphorylation events of the cell that will ultimately generate oncogenesis. We are continuously expanding the tool from lysine to other amino acid side chains. For example, most recently, we reported the photo-caged aspartic acid. Now we can activate aspartic acid-dependent enzyme, but due to the time limit, I will skip the details for this amino acid. And most recently, we also reported the chemical decay of tryptophan. This amino acid is hard to decay because the indole side chain is hard to build a caging to have a natural amino acid to incorporate, recognize this amino acid. So we built a very small venome caged indole to disrupt this hydrogen bonding of this nitrogen on the indole, as well as a peptide stacking feature, so that we can sort of lock or the cage this tryptophan. And upon this desired reaction, it will remove this venome and regenerate a free tryptophan, and as well as the tryptophan containing proteins. And I'll also skip the details, but it just shows that lots of the tryptophan-dependent protein, for example, the hydrogen bonding, metal chelation, as well as the pi-cation stacking feature, proteins can be caged and decaged, and they are therefore studied in situ inside a cell. And we're also expanding the side chain, in addition to the side chain decaging, we are developing a computer-aided proximity decaging strategy where instead of caging the specific side chain, we cage the whole pocket. And after activation of the whole pocket, we can activate the protein regardless of its active site residue. And this happened, assisted by a computer-aided algorithm and in collaboration with Professor Chu Wang. And also to make a long story short, we show that this is a general strategy by blockages the substrate access. We show that this is working whole panel of enzyme, as long as you have a structure of the protein, we can do this activation in situ. And we show also a temporary resolve the case to capture the protein, protease substrates by temporarily activate the protease, for example, caspase 3, and look for the newly generated N-terminal in a temporary resolved fashion. We uncovered many new, proteolysis substrates in situ in live cells. And most recently, we are expanding this tyrosine caged amino acid for the proximity decaging from photo decaging to chemical decaging. We managed to have this transcyclic octene caged tyrosine activated with a TCO, and then we can activate this in living animals where the light has penetration issues, but we show that we not only can activate the protein enzyme activity, but also protein interaction interface. So finally, I will finish by the last work I will show you is the new generation of covalent reaction called the, let me show you the reaction first. This is sulfex chemistry, where this sulfex reaction, if we incorporate this amino acid into protein, it can form a covalent bond with neighboring proteins. So we have a covalent linkage. But instead of Professor Lei Wang and the UCSF has done lots of work on this. But instead of the covalent inhibitor, we hope to find a degrader. We know that lots of degraders have been developed by PROTECT strategy to degrade intracellular proteins. But for membrane proteins, it's hard to use this PROTAC strategy. And there have been recently, like the LITEC strategy have been reported to degrade membrane proteins. And we developed called GluTAC, where the nanobody will bind to the cell surface antigen. And as I mentioned, this covalent linkage can be have been realized by this FSY chemistry. And this whole complex, because this nanobody also bear on the cell penetrating peptide, so this whole complex will be internalized to lysosome for degradation. And we show this is indeed the case by incorporation into nanobody, and indeed it can cross-link as well as internalize the protein for degradation. And if this occurred on cancer cells PD-L1, the receptor, then the surrounding T cell will be activated for the tumor suppression. So to summarize, we have been systematically developing bioorthogonal, genetically encoded bioorthogonal reaction to leveraging the GCE method and the bioorthogonal reactions. In particular, we are systematically unleashing the power of bond cleavage chemistry inside the living cells. These reactions are now we have been pushing them into intracellular protein, even in living animals. And with these tools, we hope to address questions to study the spatial-temporally resolved proteome. And I have shown you a few cases there, the temporally resolved proteomics will help to identify the substrates, but we're also having developing tools to study the spatially resolved proteomics tool to study the subcellular proteome. I hope I can give you more details in the future. With that, I'd like to finish up by thanking my guru, my collaborator, funding agency, and finally, thank you for your attention.
Thank you, Dr. Chen. That was really wonderful. Fantastic science. We have a couple of minutes that we can open up the floor to any questions from the audience. Suppose I can ask a quick question. It sounds you've got so you had a section there on the chemical deprotection with the tetrazine TCO chemistry, and then you followed it up with sort of photo deprotection of lysine analogs. And I was just curious to hear, it sounded like you were saying that there was maybe a preference for the photo decaging chemistry because it was faster. Is that true compared to the chemical decaging? Are there any other pros and cons of chemical decaging versus the photo deprotection chemistries that work well for you?
Yes. Thank you for the nice question. Yeah, we've been developing both chemical decaging as well as the photo decaging. There are pros and cons for each method. For photo decaging, we utilize that mostly for really rapid activation of enzyme of interest. For example, kinases. As I showed that the phosphorylation occurs in seconds.
Yeah.
And this chemical decaging can only the most efficient way in our hand also work on minutes scale or even 10-minute scale. So those are not able to study the really fast events like the phosphorylation. However, chemical decaging's pros one is that it works in living animal or live tissue where the light have a particular UV light have a penetration issue. And another thing is that the chemical decaging has higher biocompatibility. So UV light certainly still has some damage to the cell, whereas the chemical decaging, in particular, this tetrazine-based decaging strategy are really bio-friendly. And also for the time resolved studies, the chemical decaging can also work for proteins as well as some other enzymes that, for example, the caspase 3 I showed you, although we use the photo activation, chemical decaging works well because it takes like 30 minutes before we do the substrate identification, and this chemical decaging certainly meets that criteria.
Yeah, very good, very nice.
I had a quick question, Rick, if Peng's got us another second.
Yeah, go for it.
I didn't quite catch the tryptophan protection decaging chemistry. What were the R groups there that were being used there? And I'm just trying to think of size and time frame for decaging of the tryptophan work.
Sorry, the R. What do you mean by R?
I guess I wasn't sure what the new chemical structures that you were encoding were. I didn't catch. Oh, is it just the?
Oh, sorry. This Ryan can see this is a
the vinyl group. Okay.
Vinyltryptophan. Yeah, this is just a double bond, but this one can be decay rearranged by this tyrosine for rearrangement and elimination.
Dude, what is the time frame for that? I love the size of that protecting group. It's so beautifully small, but I wasn't sure how quickly that would work for you.
No, it doesn't work too quickly. The problem is that we show that for lysine we use a tyrosine because the stream promoted works certainly faster than this vinyl. But this place does not allow us to put a bulky one for incorporation. So we put this, but we optimize. I didn't have the details, but you can see in our paper that we optimize this R1, R2 position to make it fairly fast, but not as fast as those TCO promoted one.
Great, thank you.
Great, thanks, Ryan.
Yeah.
Chris, do you have a question?
Yes, I do. On one slide, you showed, I believe it was a kinase pathway, a gel, and my question was, upon activation of the initial kinase, did you see that you just passed the slide?
This one?
Not this one. This one, yes. So the question is, upon activation of the pathway, did you see any rate limiting steps or pathway expansions that you didn't expect, being able that you're able to do this so precisely?
Sorry, I didn't get your question. Can you see?
So, like for example, did different protein species downstream build up differently than you might expect from the literature, or did you see alternate activation pathways based on the literature? Now that you're able to activate the pathway with the UV light.
Oh yes, we saw lots of new things indeed, and but we have to verify that before we show this. Some of them are artifact or the real bona fide substrates. For example, for this case, this is really a well-established NAPK pathway, so we know exactly. We activated RAF, and we know exactly MAC is a direct substrate, whereas ERK is indirect substrate, and this can be recapitulated in our assay. But for us to study those, we have to verify in few criteria before we can go ahead to show this pathway can be activated. For example, shown here, the left bottom are the in vitro assay. We show that SAR can indeed activate these, can phosphorylate these new substrates. And then before we go ahead to verify the pathways. I'm not sure if I answered your question.
Thank you. Very nice work.
Thank you.
Great. Well, let's all